Meine liebe Herren und Damen, es freut mich. Pardon me, I did not remember that most of you do not speak German. So I will speak in English, but you must excuse my lapses. Because of my accent, I occasionally entschuldigen bitte. Now, I'm Robert Koch, and I'm the director of the Institute for Infectious Diseases in Berlin, and I have been so since 1891. Now let me describe to you a little bit about my early life before my discoveries regarding anthrax, cholera, and TB. I was born in the Clausthal in the Harz Mountains of Germany on December 11th, 1841, and I was the third child in a family of 13. My father was a mining engineer, and my mother, well, with 13 children, she was, you would call her today, a domestic engineer. A little joke. As a small boy, I liked to collect plants, beetles, caterpillars, butterflies, and minerals. And we had some chickens, and a cow, and a pig, and a horse, and I took care of these. I also had rabbits and guinea pigs as pets, and I learned to play the piano and the zither. After completing gymnasium, equivalent to your high school, I went to the university at Göttingen, and I began to study biology and mathematics. But after the first year, I switched to medicine. Sometimes the research at Göttingen was requiring experimental animals, and this meant me. And one time, I had to eat a pound of asparagus at a single sitting, and another time, I ate half a pound of butter each day for 5 days, and this made me so sick that it ended the experiment. And I completed my degree in medicine in 1866, and my first job was as a house physician at the Langenhagen Retarded Children's Home. And while working there, I married my sweetheart. Her name was Emmy, and soon we had a daughter named Gertrude. Our Prime Minister, Otto von Bismarck, was committed to making a united German state, and national pride provoked my 3 brothers and I to join the Prussian army when the Franco-Prussian War broke out in 1870. Now, because of my eyesight, my poor eyesight even then, I did not serve at the front. So I worked in a field hospital. In less than 6 months, we had defeated France. The French had to give up Alsace, part of Lorraine, and there was a high indemnity to be paid. Prussia became the greatest military power in Europe, and the French were humiliated. I've never understood why the French resented us. We won the war fair and square, not through cunning, but through our military superiority. Oh, I disliked the French then, and I still do, especially that narr Louis Pasteur. And at the end of the war, I left the army and became a district physician in Holstein in Polish Prussia, where I vaccinated children, I developed public health measures, and reported on epidemics. The work was extremely boring. Not only that, the pay was poor. So I had to have a private practice to supplement my income. In the little village where I began to practice medicine, there was a very serious disease of sheep. It was called anthrax. It was a killer. And when I began to study anthrax, I wondered, was this a disease caused by microorganisms? Or did the disease simply create a favorable environment in which these organisms could grow? In short, do the bacteria cause putrefaction or vice versa? And now I want to continue this lecture in the lecture room. So I bid you entschuldigen, I bid you goodbye briefly, and we pick up this lecture in the lecture room. Vielen Dank. Und Anthrax in this case, was a killer of these sheep. And I became interested in the cause of this disease. And the question that arose, was this disease caused by a microorganism, or the disease simply create a favorable environment for the microorganisms to grow? In short, do the bacteria cause the putrefaction which is associated with this disease, or is it vice versa? Now, when I began this research, it was known that microorganisms were associated with a few diseases: childbed fever, relapsing fever. But with anthrax, the situation was really very confusing. Sometimes the sick animals had their blood contained a rod-shaped bacterium, but sometimes there was no bacteria in the blood. And anthrax was also associated with certain weather conditions and soil conditions. And there was no obvious means of transmission from sheep to sheep of this disease. And the doctors and the farmers in this region, they began to ask themselves, how could this disease be caused by a microbe? Now, occasionally, as some of you know, people can come in contact with anthrax, and they can come down with the disease. And I'd like to show you at least one case of a woman with anthrax. And this is the form of anthrax that is the skin form. And as you can see, this woman, can you focus the picture just a little bit? Better. Oh, I think it's going in the wrong direction. Oh, she's getting better. Better. Besser, besser, besser. Ganz gut. This woman punctured her skin with a sliver of bone while she was cleaning the meat, and she injected herself with these microorganisms. And you see that the discoloration of the skin here is what color? Schwarz, right? Black. And this disease takes its name from the black color of the skin that the person has when they're infected. And the black color in Greek, or maybe it's Latin, I don't remember now, is called anthracis. And anthracis, everybody knows that there are two kinds of coal, right? There's soft coal and hard coal. And the name for the hard coal is called, was haben Sie gesagt? Anthracite, right? And anthracis, the stem for anthrax, equals coal, and coal is what color? Black. And that gives the disease its name. Okay. Now, first of all, one needs a method to study these bacteria. First, you have to isolate the bacteria. Then you have to grow them, and free of any other kind of microorganism. And then you have to introduce these bacteria that you've grown, essentially pure, into another animal and cause the disease. Now, when I began this work, my laboratory was simply a very small room to the side of the house. It was next to my patient treatment room, and it had a sink, it had a workbench, and an incubator. It was really a very crude laboratory. And my wife, Emmy, about whom I spoke, gave me for my 28th birthday a great gift. And this is the present that she gave me. She gave me this wonderful microscope. Schön, natürlich. A good brass microscope with a magnification of maybe 1,000 times. And this microscope enabled me to examine tissues and extracts from the animals. And I used all of my money to carry out these experiments. I paid for my own research. Now, to study anthrax, I developed a method for growing these bacteria and allowed them to produce spores. Let me show you what the bacteria look like. I'm going to make you a little diagram. So the shape of the bacteria is like this. It's a rod, and we call these bacilli, richtig, because the shape. And when these grow in the culture, eventually they develop a resistant wall, sort of like a seed. And this is called the spore. Okay, now, what we did was we took a glass cover slip like this. We take a glass cover slip. This is larger than a cover slip that I can put under a microscope, but I want you to see it clearly. On this cover slip, we put a drop of spleen from an infected animal. And then we want to observe this under the microscope. Well, we could cover this up and put it under a microscope and look at it. So here's the drop, okay? Now, you know what's going to happen if we leave this for a long time, maybe some days, in order to produce these spores. What will happen to that little drop if I left it like this? It would dry out, right? So we needed a method to observe this drop from the infected spleen for days. And I invented something that would work. And this is called the hanging drop method. So here is a slide, microscope slide, and it has a cavity in it. We take this drop like this, and we put it on this, like this. And now the drop is hanging, right? And it's sealed off from the outside, and no evaporation can take place. And we can watch this little drop under the microscope for many days. Brilliant, brilliant. Simple, but brilliant. And this hanging drop enabled us to see that the spores develop from the bacilli. It took sometimes 10 hours, sometimes 15 hours, sometimes a few days to produce these spores. And when you add fresh blood to the spores, what happens is they become bacilli again. So these are the dividing forms, like this, and these are the resistant spore-like forms that we assumed would cause the infection. Now, I caught some mice in the barn. They were the only animals I had available for this research. I fed the mice infected spleen, and what do you think happened? Gar nichts, nothing. But if I took some spores like these, and I sterilized using a flame, a little sort of the equivalent of a splinter or a toothpick, took a little bit of the infected spleen, put it into the mouse, broke the skin, then the mouse came down with anthrax. So I concluded from this little experiment that the spores must infect the animal when they enter the wound. By way of this dust or whatever schmutz is in the environment that contains these spores, and that is the way the animal becomes infected. It's just like HIV in this sense. Now, so how did the animals become infected? Their skin is abraded, scratched, and they come in contact with the spores, and this provides access for the animal to become parasitized by these bacteria. Now, of course, you know that the spores, if you have spores and they are microscopic, that you can take these dry spores, which are really very resistant, and if you inhale these spores, you can also become infected with anthrax. In fact, people who used to separate the wool from the sheep would become infected by inhaling the spores that were contained on the wool. This is called wool sorter's disease. Wool sorter's disease. And of course, today, you can take anthrax and you can weaponize it by making an aerosol of these spores. You can send it through the mail, and the person who opens this up and breathes in can become infected, and in many cases, the people die of this. Now, anthrax is caused only by this bacterium, and this bacterium's name is Bacillus anthracis. And Bacillus anthracis can produce spores. The bacilli themselves are only viable. They only live if for a short time under adverse conditions, but the spores can last for a very long time. And they can be held for years, and they can germinate and produce the bacilli. And you can prevent spore formation by lowering the temperature below 15 degrees centigrade, or you can do it by removing air. So this process requires air. Now, I was recognized for this work by our great Prussian government, and in 1880 I was appointed to the Imperial Health Office in Berlin. And I was provided with a much better laboratory. I didn't have to pay for the research materials out of my own pocket. The government now paid this. And this is when I really began my great research work. Now, in my opinion, the experiments that I've described to you and others gave conclusive proof that anthrax is caused by a bacillus, and is the only cause of anthrax. And then this Frenchman, what is his name again? Oh, Louis Pasteur. He came along. He said that he filtered anthrax blood through clay, and he inoculated the remains, and he claimed that he had made a great discovery, but actually he had achieved exactly the same results that I had already published on. And in fact, some of his work confused the issue. The matter was already fully resolved when I began my work and when I published the work. Anthrax bacilli cause this disease. Pasteur's experiments simply were old evidence in a new form. But he wanted to take credit that he had made this discovery, and being French, of course, there was this great competition between the two nations. Only a few, I want to tell you this, only a few of Pasteur's beliefs about anthrax are new, and those which are new are false. But enough of this guy Pasteur. Now, let me get back to my early years in Berlin, where I tried to grow bacteria using a liquid medium. So here are some little bacteria. That's what I say. But the bacteria, they don't want to move. Okay, so we've got this mixture of bacteria in liquid culture, right? Two kinds of bacteria. And they're swimming in this medium. You know what swimming is like, right? Okay, so they're swimming in here. And we want to separate these two kinds of bacteria from one another. And how would you do this with this liquid culture? Not very easy, right? Because they swim together. So in order to separate these bacteria in the liquid culture, let's say they're growing in meat broth or chicken soup or some other nutrient medium, what we wanted to do was to separate these. And the way to do this is to smear this out on a cooked potato. And when you smear it out on a cooked potato on a solid medium, you can take a little loop like this from the cut surface of the potato, and you can pick these little bacteria off, because what happens is the bacteria divide. So suppose we have two kinds of bacteria, blue ones, and we have black ones. They're swimming together. But if you spread them out on a solid surface, they begin to divide. And they divide this way, this way, this way. Now the individual bacterium is invisible to the naked eye, but the product of all of this division, which we call what? Asexual product of a division is called a clone, and this clump is called a colony. And this colony is visible to the naked eye. And the same thing happens here with the blue ones. And you can see that their colonies are separated on this solid medium. Now, potato is really not the best sort of material, so we decided to do this using gelatin. So now we have it on a solid surface with gelatin. You know what gelatin is. We eat it, right? What is gelatin? It is called commercially Jello. And Jello, as you know, if you've ever made Jello, you heat it up to almost boiling temperature, and then you let it sort of cool down. And at room temperature, it's still sort of semi-solid. At body temperature, it's very liquid. But you could separate some of the bacteria using gelatin, gelatin plus broth. But it's not very effective if you want to grow microbes that grow in us, because the gelatin is liquid. And so we have the same problem that I just shown you. They'll be swimming in this. So what we needed was a solid medium that would not be liquid at body temperature. And thanks to a young woman who worked in the laboratory, actually the husband, the lady's name was Fanny Hess. And Fanny Hess was actually born in New Jersey, but she married one of my associates. His name was Walter Hess. And she had learned that in Indonesia, that in order to solidify jellies, Indonesia, of course, having warm temperatures, that they used a material from a Japanese seaweed that would make the jellies solid. And this material we call agar. Not only that, if you now put the agar into the medium, you can now streak them out, and you get separate colonies on this agar medium. And the agar doesn't influence the growth because it's inert. It's a polysaccharide. Now, one other important development, also developed by one of my associates. His name was Petri, Richard Petri. And Richard Petri made a great innovation. If we had agar like this, and we exposed it to air, what would happen to the agar? What happens with jello if you leave it open to the air? It dries out, right? So what we needed was a little dish in which the agar could be poured, and then if we have it like a pill capsule, so here's a large one, and what we did was we have this pill capsule made out of glass. There's a top and a bottom. You put your agar in here, and you take your needle with your beautiful pink needle here, and you streak out from whatever source you think you have the bacteria, and you go like this on the solid medium, and eventually what happens is you can pick the colonies and you can have pure cultures. And this little dish, what do we call this dish? Petri dish. Very good. And here is a real Petri dish. I think I have a few here. So here are Petri dishes. We now have them in plastic. And these bacteria here are not anthrax because they have the wrong color. This color is red. This is Serratia. So you can now look at this, keep it around for a long time, observe the colonies. You can see the little colonies here. I'll pass this around. And you can see that you can now pick each one of these when you want them. Of course, you have to open the dish, but it never dries out. Now, what we've done here is here's another different kind of bacteria. You can see that the color is slightly different. Now, suppose we mix these two together. We want to separate them. So what we do is we streak them out until we have pink ones and white ones. And then they separate, and then you can grow them up separately. So here are the Petri dishes. Don't eat them. They're poison. Just look. Okay, so now we have agar and we have Petri dishes. Now these simple inventions that we made in our laboratory in Berlin were announced at the 7th International Medical Congress in 1881. And even Pasteur came forward and said, this is a great achievement. Of course, he said it in French, and I don't do very well in French. But in any case, the translation is, good work, Koch. Now, after this, I left the problem of anthrax, and I tried using the same techniques to study tuberculosis. Tuberculosis, you know, is a disease of the lungs, but other organs as well, but mostly the lungs. And we believed that the cause of this disease was a bacterium. It causes a wasting of the individual. We call this consumption, because the person seems to be eating up his own body when he's infected. Now, at first, we had to see these bacteria under the microscope, and we used a dye to stain them, because most bacteria are colorless under the microscope. And the dye that we used was an aniline dye. One of the aniline dyes is called methylene blue. So if we had a bacterium that's ordinarily colorless, we could color it with this dye, like so. The colonies would actually still be colorless, but under the microscope they would look blue. When methylene blue or these aniline dyes are derived from coal tar. And in Germany at this time, we produced all sorts of products from coal tar. In fact, the whole development of organic chemistry, which is the chemistry of carbon compounds, is developed by using as a starting material coal tar, produces aniline, and then you can produce all kinds of different stains. What's ironic, isn't it, that the first bacterium that we talked about is named after coal, and now we use coal tar to stain these other bacteria. Now, suppose we find in a person who has tuberculosis a bacterium, and we can stain it with methylene blue. We cannot simultaneously conclude that because this individual has these bacteria, that the bacteria are the cause of this disease. Can we? No. What we need to do is to establish what we now call today, after me, Koch's postulates. When Koch's postulates are very simple in concept, but sometimes very difficult to prove. So we have a diseased animal. And when we have this diseased animal, we obtain from this animal the microorganism or microorganisms. When this microorganism or microorganisms have to be grown in culture free of any other organism. So what we call this pure culture. So for example, let's say we had 3 different kinds of bacteria. If we grow them separately, red, blue, yellow, these would be pure culture, right? Or this would be a pure culture. Now, part 3. These microorganisms that are grown in pure culture are inoculated into a clean or healthy animal. And what happens? This animal shows the same signs of this disease. So the disease is produced. And then, finally, what you have in this is you have to isolate again the same microorganism that you obtained here, grown in pure culture. If these postulates are satisfied, then you have proof positive that the causative agent of this disease is in fact the agent responsible for producing the disease. So I employed these postulates and the techniques I've described to you for tuberculosis. I grew these bacteria that I obtained from a patient from the sputum of this patient. What do we call sputum? Spit. Not schnot. Spit. Sputum. I will spit on you, and then you will know what sputum is. There used to be a sign in the New York subway system. It used to say, if you expect to rate, don't expectorate. Because when you spit, that's called expectoration. So it's a play on words. The New Yorkers are very, very clever, especially in the subways. Okay, so what we did was we grew these tubercle organisms, and this is another bacillus. It's called the tubercle bacillus. When this bacterium was obtained from a patient, we grew it in pure culture. We inoculated it into guinea pigs, and the disease is reproduced in these guinea pigs. And we could transmit this disease, unlike anthrax, by spit, by sputum. Now, even if these microorganisms are dried, they became infected. It's very difficult to become infected with tuberculosis by injection. So unlike anthrax with the cuts and the wounds, here is a disease that spread from individual to individual by droplet infection. Now, you might ask, what the hell good is this, right? Well, it's a gain for science. If one has shown that a disease is caused by a particular agent, a parasite of sorts, now we can use this to study other diseases, and we can use it to control infections. So this may be a very, very useful tool, not only prevention, but we can use it in diagnosis. Now you may recall, those of you who attended the Monday video, that tuberculosis was considered a disease of conditions such as poverty, overcrowding, poor hygiene. But that's not the cause of this disease. The disease is caused by a bacterium. And now that we know this, we can investigate it further, and we can understand how this disease is produced in these animals. And as we have said, you might develop a cure for this disease. And we also can develop a way of controlling this disease. For example, if sputum is one of the means for transmitting this disease, then we might disinfect sputum, and you can then eliminate transmission of this disease. We can make the sputum harmless. And in addition, we can disinfect objects that come in contact with this sputum. For example, clothes, food, utensils, anything that the person who has tuberculosis would come in contact with the sputum would be a means of transmission, and you can eliminate transmission by either washing, sterilizing, disinfecting these kinds of objects. Now, another source of tuberculosis is the tuberculosis related to human tuberculosis, and that is tuberculosis of domesticated animals. In fact, identical with human tuberculosis is tuberculosis in cows, bovine TB, and it's transmissible to humans in both the milk and the meat of these diseased animals. And the way we treat milk in order to eliminate this disease, and I hate to admit it, what do we do to milk? Ooh, I hate that verb, that pasteurization, but it turns out that it is in fact one means of controlling bovine TB from becoming human TB. Now, my success with tuberculosis was recognized worldwide, and in 1905, I was awarded the Nobel Prize for this work. And I was called upon after the work on tuberculosis to study cholera, and I discovered the causative agent of cholera. Some of you know that this is, what's the name of it again? Vibrio cholerae. And this has a slightly different shape. It looks like this. It has a comma shape, but it is another bacillus, and I was able to identify this bacterium when there was an outbreak of cholera in Egypt in 1883. And we grew this bacterium in the way I described to you. And we found that the bacilli of Vibrio cholerae die if you dry them. I think I have TB. So if it dries out, this Vibrio, it is no longer able to transmit the disease. So it has to be in a liquid form. And this means that unlike tuberculosis, cholera is spread by water, contaminated water passed out from the feces where the bacteria live, and it's the excrement, or sometimes we might call it night soil. You remember night soil? That is the means of conveyance of this disease, but it also can be contaminated food. It can be contaminated utensils. Insects can even, you know, when the flies drop on the excrement, they have their feet. They can also transmit these bacteria from one person to another, and you can get cholera this way. If you have low temperature, below 17 degrees centigrade, the Vibrio cholera do not develop. And in places where there are epidemics, there are individuals that develop immunity. And so some of these outbreaks of cholera sort of dampen out because of immunity of the people. So cholera epidemics may stop on their own. And we were able to produce cholera-like symptoms in guinea pigs by injecting these comma-shaped bacilli into the intestines of these guinea pigs. In 1890, I had the honor of delivering the keynote address at the 10th International Congress of Medicine. This was a very important congress. It was held in Berlin. It was a means of establishing further the national pride of science in Germany. And it was being held in an auditorium that held 8,000 people. This was a gigantic auditorium, and it was constructed as if it were the temple of Zeus. So this was a very elaborate auditorium with lots of people, 8,000. That's almost half the number of students here at UCR. Imagine all of them crowded in this one classroom to listen to me speak. And the German government at this time had pressured me to announce a preliminary discovery that I had made with regard to tuberculosis. I had found that some of these bacteria had a kind of waxy coat. And you could extract this material from the tubercle bacillus, and we call this material tuberculin. And in preliminary experiments with these extracts from the tubercle bacillus, it showed that in guinea pigs, and guinea pigs are highly susceptible to TB, that these guinea pigs were cured of this disease. And the government said, this is a great discovery, you must announce this at this Congress. And if you do so, we will give you a bigger and better laboratory. So I went to the podium, as I'm doing now, and I said, blah, 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 blah. Of course, it was blah, blah in German. And eventually, I said in German, my discovery of the tubercle bacillus led me to seek substances that would be therapeutically useful against TB. And I tested many, many substances that might have an influence on these cultures of tubercle bacilli. And even small doses of many substances slowed the growth of these tubercle bacilli. But the most effective one was this material which we call tuberculin. Well, so after many failures, I finally succeeded in arresting the growth of these tubercle bacilli. And in addition, I was able to do this with infected guinea pigs. And I concluded my speech by saying, it is possible to render harmless the pathogenic bacteria that are found in a living body, and to do this without any disadvantage to the body. The material tuberculin For example, we discovered a us. Und tetanus antitoxin was also discovered by us. My colleagues were von Behring, Welch, Kitasato, Eichmann, Wasserman, Löffler, and Pearl Ehrlich. On this little dish which I have shown you, this was discovered also in my laboratory. What's this dish's name? Petri, right. It was a wonderful life in Berlin in those days. But in recent years, I've become more and more depressed. And we had no Zoloft in those days. Or Paxil. And so, one of the reasons for my depression was one of my students, von Behring. He won the Nobel Prize before I did. And he even patented the diphtheria antitoxin and made lots of money as a result of this. But the most depressing part was this announcement that I've just told you about, tuberculin. It turned out in subsequent experiments and trials on humans that tuberculin was a poison. It was not a cure. More people who received tuberculin died from this administration of this substance than were cured. So the announcement, with great fanfare under great pressure by the government, turned out to be a personal disaster for me. But while I was at my lowest point, I was sitting for an official portrait, and I saw on the wall of this artist studio a portrait of a young actress. She was quite a good-looking young woman, and since my wife Emmy and I were no longer living together, I decided that I would pursue this lovely young woman, and we married shortly thereafter. I was 50 and she was 21. This was a May-December marriage, but it was a very conservative environment in Germany, and I became, and my wife became, ostracized from society in Germany. We became social outcasts. So we traveled extensively. Frequently I was asked to look into infectious diseases and determine the causes of these diseases. For example, malaria was a disease that I was called upon, African sleeping sickness, West Coast fever. But in none of these cases was I able to identify the causative agent for these diseases. And the reason is, of course, that these are not bacteria diseases. These are diseases that are much more difficult to grow in pure culture using these techniques that I've described to you. And so I failed. I didn't understand why at the time, why I failed, but I can take some consolation in the fact that neither did Louis Pasteur, that miserable Frenchman. And so I conclude by saying, auf Wiedersehen. Fertig für heute.